Listen to me. Thank you. So, welcome today to the 3-minute thesis competition. Now, this is the first event that the University of Cambridge have run campus-wide. It's amazing. If you haven't heard about the competition before, I really encourage you to, particularly if you're from an international institution and you're maybe here for a year, I'd really encourage you to bring it to other campuses. Now, if you're not aware of the event, and you're here going, well, it sounds like a great concept, tell me more. Essentially, it's about communicating research and communicating it in an engaging way so that you actually engage an audience. Now, there's two sides to that. The general public need to know what's coming out of research so they can respond to it. The second part, people who normally write grants aren't specialists. This means you can communicate in a way that you're actually writing those grants will be affected. Actually presenting your materials will get you the money, which is key, really, if we want to continue. So, essentially, we have some amazingly talented people here tonight. Now, they are the results of 4 heats we ran in April. This spanned an amazing range of departments and colleges. Now, just to give you a sample of those, here are some of the departments represented tonight. We have a huge range: plant sciences, chemistry, history of art, physics, engineering, right through to pathology and the police department. There's an amazing range there. And that represents, we even had polar studies in the heats. It got really, really diverse. And that represents the huge chunk and diversity of students we have here at Cambridge, which you really need to showcase more often. In terms of the colleges represented here tonight, in terms of heats, we pretty much had everyone. But the colleges, we have Magdalen, Newnham, have 2. St. John's, Churchill, Corpus Christi, again with 2. Homerton, Jesus, Mary Edwards, Clare, and Emmanuel. There's a really great range of people involved tonight, and I'm really excited you're here to hear them. Now, we should just mention our judges. We have an amazing range of people here tonight, and if you want to look up their CVs, please do so, but if you do have chronic, oh God, my career syndrome afterwards, don't blame me. So, we have Fiona Carrott, Professor at the Department of Medical Genetics. Please look up her CV or just follow her at Darwin College, either one. Professor Frank Carey, Master of Christ College and Research Fellow of Christ College, Professor of the Mathematics Department as well, Mathematics of Systems. You have a fantastic career here, and I'm not going to go into it, as do most of you. So again, please have a look, and I should just say they will be here afterwards, so if you want to grab them and learn things, please do. Professor Anna Vignoles from my department, Professor of Education, Director of Research at Department of Education and Fellow of Jesus College. Professor Jeremy Sanders, Professor of Chemistry, Department of Chemistry, and Pro Vice Chancellor for Institutional Affairs. So please have a conversation with him as well. And Professor Graham Burton is Professor of Reproductive Biology at Department of Physiology and the Mary Marshall and Arthur Walton Professor of Physiology. I'm going to start again. This happens. You get through so much content, you're just like, wait, no, breathe. So, Mary Marshall and Arthur Walton Professor of the Physiology of Reproduction. There's going to be many of those tonight where I just go, okay, no, breathe, 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 take it all in. So, amazing range of people. Now, I should just mention, if you can turn your phone off before we start anything, I know it's something everyone says is annoying, but it is key because otherwise, well, I'm not going to take responsibility if one of the contestants comes up and just gives you evils, basically. Please turn them off. So, with that, I should just mention after this event, there will be wine or juice if you're preferable and you're through your thesis right now at the GU. There are also snacks which are amazing and I had to stop myself. So please come to the GU afterwards. It's just a 2-3 minute walk up there and follow me because I'll be the one at the snacks. Now, kicking off, essentially the way this is going to work is there are going to be 3 lots of 4 participants. Now that gives you a bit of breathing space to go, okay, collect thoughts, which I need previously. We're going to introduce the first 4 for you now. Now, we've got Vi, doctoral student from Magdalen, who's doing plant sciences. Next, we will have Jackie from Newnham, doing a master's in chemistry. After that, we will have Christina from St. John's, who's doing her master's in history of art. And the last one in this group of 4 will be Frederick, who's studying physics and at Churchill. Okay, now kicking off with the first one. You are so brave. Now, Vi is of course at Magdalen, and his faculty is plant sciences, and his title is, deep breath from me, Algae-Bacteria Symbiosis, using molecular biology to improve algae cultivation for biofuel. Over to you.
Good evening, ladies and gentlemen. Faced with the twin threats of climate change and an unstable energy market, research into alternative energies has never been more important. One potential source of alternative energy is algae. Algae are able to take sunlight and convert it into, or use sunlight rather, as an energy source to convert carbon dioxide and water into biomolecules such as glucose, which algae can then metabolize using their own metabolism to produce molecules such as lipids. These lipids can then be directly used as biofuels. This was discovered about 10 years ago, and many people believed we'd have large-scale algal ponds growing and producing tons of biofuel. Unfortunately, one of the major hindrances to achieving this goal is contamination of algal ponds. And so my research group here at Cambridge studied the metabolism of algae with the aim of understanding how exactly is the best strategy to grow them. A few years ago, we made a seminal discovery in which we showed that of over 300 algae that we know, over half of them require vitamin B12. This was published in the journal Nature, and one example of such an alga is this one shown over here, where if you deprive it of vitamin B12, you get a clear, transparent liquid media, meaning they don't grow, but if you provide it in the media, they grow very happily. The second question we asked is, so where in the natural world would vitamin B12 come from? And it turns out that only bacteria can make vitamin B12. And so this led to the idea of building synthetic ecosystems, basically co-cultivating algae with beneficial bacteria, effectively probiotics for algae. And the system we then developed in our lab was a co-culture system with this alga shown over here called Lobomonas rostrata, and it grows with a bacterium called Mesorhizobium lotti. Tricky names, but you don't need to remember them. The key thing to remember is that they can coexist together. And the important thing is that pathogens that were causing the contamination can't fill the niches once they're occupied by the good guys. So my research then focuses using microscope techniques, so high-resolution microscopy, genetic analysis, and what I'm trying to understand is exactly how this co-culture functions. And through understanding this, the aim is then to build a microbial consortia of several different bacterial species that support growth of algae on a large scale. And with that in mind, we hope to then realize this dream of having large ponds of algae producing tons of biofuel and a more stable climate, more stable energy market. Thank you very much.
Well done. Thank you, Vi. Brilliant stuff. During this, particularly with the science presentations, I suppose it's the intent of this competition, but you're probably going to feel like you wish you had these people in science as your teacher when you were at school. You'd probably be studying science now. So, great stuff. Now, next up we have Jackie from Newnham. Now, Jackie is a master's student from the chemistry department, and her title is Using Chaperone Proteins to Inhibit Myeloid Fibril Formation in Alzheimer's Disease. I'm getting much better at this. Over to you, Jackie.
If you picture a human cell as a city, then proteins are its citizens. They perform a wide variety of jobs. Some defend the cell, some are messengers, and still others regulate their fellow proteins. A protein is born as a linear chain of molecules, which then folds into a specific three-dimensional shape. This is a complicated process, but the fold is critical for the protein to function properly. And so the cell has evolved a vast maintenance system to ensure that proteins fold correctly and are repaired when they don't. However, when this maintenance system breaks down, diseases caused by the resultant protein misfolding can emerge. In these disorders, misfolded proteins aren't repaired, but instead they clump together into plaques called amyloid fibrils. When such misfolding and aggregation occurs in the brain, diseases such as dementia, and most commonly Alzheimer's, can result. In this top image of a brain slice from an Alzheimer's patient, the brown clumps are the amyloid plaques. This aggregation is toxic to neurons, killing them and causing cognitive decline. In my research, I focused on one potential way to inhibit amyloid fibril formation. Interestingly, this inhibitor isn't a drug or any external treatment at all. In fact, it's another protein produced by the body itself called a chaperone, aptly named because its task is to help other proteins fold. So in addition to its regular job supervising normal folding and repair, chaperones are also potent inhibitors of amyloid aggregation. To see how they do this, let's take a closer look at the amyloid structure. If you were to zoom in on these brown plaques 1,000 times, you would start to see individual fibrils, like this one on the bottom. And these fibrils are notorious for causing chain reactions of aggregation. So once you have just one fibril, other damaged proteins can more quickly stick to it, accelerating the aggregation process. And here's where chaperones come in. A chaperone can recognize an amyloid fibril and stick to it, which you can see here. The chaperones are the black dots adhering along the fibril. They can then block other damaged proteins from sticking to the surface and thus delay amyloid growth. I've been studying one particular chaperone and have been able to measure both how quickly and how tightly it can bind to fibrils. And with quantitative information such as this, we'll be in a better position to inhibit amyloid fibril formation and combat neurodegenerative disorders like Alzheimer's disease by harnessing the cell's own protein policeman, its chaperones. Thank you.
Well done, Jackie. I'm from Unum. I'm just declaring that now. Well done. Okay, now next up, I'm quite happy at this. I am a humanities kid, and I'm doing okay with the science pronunciations, but I'm very much a humanities kid. I'm holding my hand up now again. Now, Christina is next from St. John's, History of Art student, and her talk is on clocks in Tudor portraits. The slide's probably a clue. Over to you.
What is the point of a painted clock? It always says the same time. What we see as its usual function is gone. So why did so many people in Tudor England have themselves painted with clocks? Just one slide, so you're going to have to take my word for that. Until now, nobody has tried to fully answer this question. The chances are you've got a clock with you, a watch, or maybe a phone. I'm currently very conscious of the clock, but most of the time we don't really think about it. For us, clocks are functional. But in Tudor England, clocks were richly symbolic. For this man, William Chester, Lord Mayor of London and successful merchant, the clock expresses a range of ideas, and I'm going to focus on 3. The first is wealth. Clocks were inaccurate and watches were worse. People didn't need them either because sundials were better and cheaper. But clocks were expensive. Whether or not Chester actually owned this clock, he wants you to think he did, because that tells you he's a wealthy man. The second meaning clocks could have is to remind you of mortality. Real clocks could be decorated with skulls, and in most clock portraits, skulls also feature. William Chester goes the whole hog and puts an entire skeleton into his portrait on the lower weight, just where it says death at hand. Clocks remind the Tudor viewer that time is running out. But clocks had other meanings. Religious texts used clocks as metaphors to describe the universe. Everything from the human body to society itself was described in terms of one wheel moving another wheel and then another. Clockwork as a chain of causes fascinated Tudor writers. But they also liked the fact that the workings on the inside of a clock were shown on the outside by the moving hands. Now, in the 16th century, religious discord split Christian Europe in half, and one of the major disagreements was over what got you into heaven. Was it a combination of believing in God and doing good things, as Catholics thought, or was it faith alone, as Protestants said? William Chester was a Protestant who believed only his faith in God would save him from hell. But for Protestants, there's an added problem. How do you know you're saved? How do you know what's going on your inside from the outside? Well, the clock metaphor comes to the rescue. Just like a clock, preachers said, where the clockwork inside moves the hand outside, you know you are saved by the good deeds that you do. But, they add, the good deeds are an effect, not a cause. The clock moves the hand doesn't move the clock. What about William Chester's soul? Death sits on one weight, on the other stands Christ after the resurrection, next to the words hope to live. Christ is on the up, he's beating death, who's sinking. The time on the clock says 1, the start of the day. Things are looking pretty good for Chester's soul. In this portrait, and in many others, the clock represents the sitter's soul. It's difficult to show beliefs visually, but here the clock is shorthand for I am a good person and I hope to go to heaven. Painted clocks can't tell you the right time, but they can tell you about the sitter's concerns. Wealth, mortality, resurrection, these are just 3 meanings that clocks could have in Tudor England. There were also others, but today I don't have time. Thank you.
That was basically 3 minutes. You were perfectly timed. Perfect. Now, finishing our last set of this 4 is Frederick. Now, Frederick is a doctoral student from Churchill, and this sounds like it could be a movie title. This is a good title. He's talking about the quantum revolution. Perfect. Over to you, Frederick.
Let me ask you 2 questions. Have you ever been so busy that you wanted to be in 2 places at the same time? And have you ever had an email that was so confidential you wanted to be absolutely sure nobody would be able to hack into it? Well, if these visions sound like science fiction, I got news for you. We've already realized them. And I will talk to you tonight about the quantum revolution. This is all based on quantum physics, which has some highly counterintuitive properties. For instance, particles can be in 2 places at once. That is illustrated over there, where we have poisonous particles which are in 2 places at once, and hence the cat is both alive and dead. Let me assure you, though, it's just a thought experiment. We can use these counterintuitive properties to build new technologies. However, there are 2 main problems: fragility and scalability. So quantum states are very fragile and easily collapse, so we need to protect them. And also, it's not enough just to have a single quantum particle. You need to have hundreds or even thousands in order to build a useful technology. In order to address these issues, the unique approach I've pursued in my PhD has been to use semiconductor chips, which are less than a centimeter long, and on which we can have hundreds or even thousands of particles of light called photons. Now, the potential for applications is enormous, so we're really talking about a revolution here. 2 examples. For instance, quantum computers can predict how proteins fold and are thus arguably the key to cancer and, as we've seen, Alzheimer's research. In addition, in quantum communication, you can mathematically prove that nobody can eavesdrop without being detected. Now, I think I've just heard a few jaws drop, so I will repeat these points. Quantum computers are likely the key to cancer and Alzheimer's research. And in quantum communication, nobody can eavesdrop without being detected, whether they're using the most old-fashioned or the most modern eavesdropping techniques. Now, if you think this is just some sort of pipe dream, I can tell you that this revolution has already begun. Swiss banks are using quantum communication, and currently the world's longest link is being installed between Beijing and Shanghai. In case you're wondering what's next in this revolution, what we're working on, well, we're still working on being able to place you into 2 positions at once. We haven't quite managed that, so it might not make it into my thesis, but hopefully we'll get there soon as well. So thank you for listening.
I was right, it was a science fiction movie. All right, so thank you to all our first 4. That was brilliant. Now, just to give everyone a chance to breathe, key, we have 4 more coming up for you, so I'll introduce them as a group now. So, first up we have Kirsten, doctoral student from Corpus Christi, from the physics department. Then we have Just, a Homerton student who is studying at the psychiatry department. Then we have, Just. You taught me how to say that, and I said I'd remember. And then we have Jane, who is a doctoral student from Jesus College doing development studies. And lastly, we have Liling Zhao, an engineering student from Murray Edwards. So, first up, we have Kristen. Now, just let me give a little bit to just introduce her topic. Okay, so her topic is going to be DNA origami. You could probably tell that, but just in case. And again, Corpus Christi and a doctoral student. So, over to you.
let me tell you a story about crocodiles in Cambridge. Not quite an urban legend, but a story about one of the latest wonders in science, about folding or about DNA and origami. You all know origami, right? Paper folding. Now, what if I told you that in my lab we are folding DNA into any shape, for instance, into the shape of this crocodile? We make not just one, but billions of nanometer-tall copies of the logo of the physics department. If one of them was the size of a real crocodile, a grain of sand, in fact, would be larger than Cambridge. For me, this still sounds like science fiction, but believe me, it is not. So nature has invented DNA to store the information of life. And today, scientists use it as a construction material. We're essentially building with the building blocks of nature. No genetics, simply architecture. So how do you fold DNA? Well, DNA is made from two strands. If they match, if the letters of the genetic alphabet match, they bind and form a rigid spiral staircase. Now imagine you have a long piece of DNA and a short piece that matches at two distant ends, say on the left and on the right. Then you can pull this long strand together in the middle and you fold it and you have something like a loop. Now imagine you have many of these short strands. You can make a crocodile or anything else. In practice, you just draw the shape you want on the computer, which then gives you a list of DNA sequences. You order them online, you mix them, and the DNA origami is ready. Cheap, biodegradable, and safe to use, unlike other forms of nanotechnology. So we have this really beautiful technique to build on the nanoscale. And what we do is we make crocodiles. Well, DNA origami right now is at the transition from proof of principle, from artistic shapes towards applications in the real world. And ideas come from the most diverse fields. I myself, for instance, I fold pipes, like small channels, from DNA origami. They look boring, a bit like this paper origami model, but they can punch holes into cells. So you should know that 50% of the drugs that we currently use target natural channels in cells. So imagine what we could do if we could create artificial channels for every patient the way we need them. We could, for instance, deliver drugs, or we could stimulate heart cells that beat too slowly, or we could kill diseased cells. Now, for now, this is a vision. But to use the words of paper origami artist Robert Lang, one day origami might even save a life. And one day, I tell you today, the same may be true for DNA origami. Thank you.
Okay, I've got to ask, when listening to that, did anyone, when they heard crocodiles in Cambridge, think, ooh, excellent story? It didn't quite go that way in the end, but brilliant stuff. Okay, now just before I introduce the next one, we are recording this so that it's going to be available for use. So can participants just be aware of the microphone, and judges, please don't judge people for sticking too close to it. So essentially, coming up we have Joost. Am I saying it properly? Joost? Joost. There we go. Now, Joost has actually brought a whole support team with him from the Netherlands, which I think is excellent. Well done. Now, Joost's presentation is going to represent the psychiatry department, and his presentation is How Do Delusions Emerge? Over to you.
For the next couple minutes, I want to talk about what I find is one of the most interesting questions in cognitive neuroscience, perhaps even science in general. That is the question about how does biology relate to subjectivity, like thoughts and beliefs? And although it's a very interesting philosophical question, it's actually very interesting for psychiatry as well. Because it is in psychiatry where we try to treat subjective symptoms like delusions and hallucinations by giving them medication that works on the biology. And it's very important to understand this relation between biology and the symptoms so we can develop better treatments for these people that suffer from psychiatric symptoms. Let me give you the example of psychosis. Psychosis is a mental state where people experience hallucinations and delusions. Yet despite decades of research, we have very little understanding of what in the brain's biology causes them to have these hallucinations and delusions. To give a relevant example, we know that a certain neurotransmitter in the brain that those neurotransmitter levels are different in people that suffer from psychotic symptoms compared to people who do not. Yet we have no understanding at all why it is that these changes in glutamate levels causes someone to believe that the MI6 is out to get them. There's an explanatory gap between the biology and the subjectivity, and my PhD aims to fill that gap. But before we get into how the delusions might emerge, we first have to get an understanding of how the healthy brain forms beliefs about the world. I'll be ambitious and try to summarize this in a couple sentences, and that is, what the brain essentially tries to do is try to predict input to the brain and try to optimize these predictions of the input by comparing it to the actual input to the brain. And the difference between the two, called the prediction error, is used as a teaching signal to update these predictions, so the next time the brain is making a prediction, it will be closer to the truth next time around. So let me give you an example. You might think I'm not a very generous person. You might expect me to give just £1 to Nepal. Instead, I give £5. There's a prediction error of £4, and that prediction error will teach you to make better predictions about my generosity next time around. So in this framework, this prediction error is key in updating beliefs about the world. So if this coding of the prediction error goes wrong, you might expect wrong beliefs to be formed about the world. And indeed, in people with psychosis, where we can measure these prediction errors during learning, we see that the coding of these prediction errors is going wrong. So might this be the missing link between biology and symptoms? Do these glutamate levels that are different in people with psychosis, do they affect the coding of these prediction errors? My aim is to address that question by measuring these glutamate levels in people that suffer from psychosis, people that are prone for developing psychosis, and healthy controls, and see how these glutamate levels affect the prediction error coding. And when we have insight in these relations, we have a better understanding of how the biology might affect the way we learn things about the world, and perhaps even delusions, and in that way we can perhaps make better treatments so fewer people have to suffer from psychosis in the future. Thank you.
Excellent, and lovely use of art there. All right, now, I love how we've kind of sorted this. Now, I just did this alphabetically. We've actually got a lovely mix because it's science, science, art, science, and the same is for this one. We're moving to development studies. Now, Jane is a doctoral student from Jesus, and her presentation is on farming for survival versus farming for gain. What makes the difference in Rwanda? Over to you.
This is Mercy and her husband, Jean de Dieu. And like about 80% of the farmers in Rwanda, the population in Rwanda, they're farmers. And they used to just farm for survival. But as a couple, they've made some big decisions recently. And now they grow rice. Traditional farming, pretty basic, without any assistance from animal power or mechanization. And the characteristics of farming for survival are that you try and grow a wide variety of crops so that whatever the weather does to you, you're going to have something to eat at the end of the season. You try not to spend or risk money. You save your seeds from last season to plant again this season. And you eat what you grow. Now, the Rwandan government, a strong government, some would say autocratic, has made a decision that the agricultural sector really needs to grow and transform. There's no new land for production, so the only way you can get any kind of transformation is to intensify for production. And that involves this transition from farming to survival for farming for profit. And the characteristics of farming for profit are, firstly, that you invest, you buy seeds, you buy fertilizer. You also grow many fewer crops with the intention of selling them and living on the cash that you get from having cultivated. And so the final thing is you're not really growing to eat. You're growing to sell and live on an income. In short, farming for profit involves investment, and that means risk. And you could do this by getting big landowners involved. You could get them to buy up all this tiny, tiny pieces of land. You could get them to hire these lovely farmers, and you would get the growth that you're looking for. But the Rwandan government has taken a slightly different approach, and they are trying to engage farmers much more directly in this transformation. And they're asking individual farmers
to invest in their own production on their own land. Now, this is a very strong top-down policy, and it's operating within a framework of support so that farmers are encouraged to grow particular crops according to location and so on. But finally, each farmer or couple has to decide. They have to decide what to do. And my research, I focus on how they decide. How do they make these decisions about money? How much do you risk? How much do you invest? And where do you get the funds when you actually have made your decision? So I use population-based statistical information, but I also do a lot of interviews. And I do these kind of participative group work exercises, and that's when I took this particular photograph in January, I think. So here's a really interesting insight. You've got a very strong top-down government policy, but actually it hinges on the individual decisions of millions of little farmers, which is a completely bottom-up reality. So I'm going to introduce you again to my farmers, Mercy and her husband, Jean de Dieu. They are farmers, but they're also investors, and they're also leading the implementation of a key government policy for the economic transformation of their country. Thank you.
Well done. I can hear your cheer squad whooping from here. I like it. Spirit. Good. Perfect. Now, next up we have Lilin Zhao from the engineering department. Now, apologies to Lilin Zhao. This is my fault. It's formatted slightly differently. But her... Oh, well done. I forgot about this. Now, we do have two people from Newnham and one from Murray Edwards, so excellent show. And her presentation is Flexible Polyhedra. Over to you.
Good evening, everyone. My name is Ayla Lijing Zhao. I'm a third-year PhD student doing structural engineering, and my research topic is flexible polyhedron. And now you might ask, what's that? Well, I think you probably should, because they are completely new objects. But it's such an old topic. For centuries, we've been kept seeing rigid polyhedra like this cube. And that was the very reason why, back in 1766, Euler, among other mathematicians, broadly conjectured that polyhedra, which are closed surfaces composed of rigid polygons, are all rigid. None of them are flexible. Moreover, since 1864, we've had Maxwell's famous structural engineering rule to define the rigidity of polyhedral frameworks, and we still think all polyhedra are rigid. However, in 1970s, a mathematician found a few polyhedra that are clearly flexible and for which Maxwell's equation is insufficient. And in my PhD work, I've optimized the flexibility of these polyhedra by developing a novel algorithm. And now my polyhedra can flex as far as 75 degrees. I know this may look like origami to you, but yes, I do have paper models and metal models of this one. And just from paper folding, based on my knowledge of this polyhedra, I also configured a polyhedra with more than one hinge, which means more than 1 degree of freedom. So overall, what I'm trying to do is to create polyhedra with more flexibility and make this very rare phenomenon common so people can see flexible polyhedra anywhere. But what's the point? Well, spatial engineers can potentially use the flexibility of polyhedra to create large deployable structures. This means we can fold and transport structures very easily on Earth or in space, whether it be solar panels wrapped around a spaceship or just a foldable foam. At the moment, I'm just exploring the theories behind all these concepts. So, so today I'm just here to advertise this new phrase, flexible polyhedra, and this highly unusual object, which I hope in time you will all be able to benefit from. So please look out for Li Jinzhao flexible polyhedra in the future. Thank you very much.
Well done. And with that, we are on to our last four. Now, that's gone really quickly. This often goes through, and the way, if you want to look at other competitions around the world, I really encourage you to. This competition came from Queensland, and as a result, I guess, they have a very strong showing, as do Melbourne, self-promoting. And the UK, we should say, the winner from today goes through to the national final. And some universities have this competition well established, and they've got some excellent entries as well. So if you're thinking of entering next year, or indeed if you're from another institution and you'd like to implement it, please have a look online, because there are some great resources. I should just say we have a camera at the back, so we hope to contribute our own at the end today. So if you want to have a look through any of these, you are free to. Okay, coming on to our last four. Feels kind of sad. We have Miguel, who is a postdoc from Corpus Christi, representing, and from the Faculty of Economics. We then have Henrika, no bias, she's from UNAM, a pathology student and doing her doctoral degree. We then have Anna from Clare, doctoral student in biology, and lastly Sophie from Emma, representing the police department with her PhD. Okay, first up is Miguel. You pumped? All right, now Miguel is from Corpus Christi, and his thesis topic is technological unemployment throughout history. Over to you.
Thank you, Kate. My research focuses on technological unemployment throughout history and what that means for the evolution of today's jobs. In my dissertation, I proposed a model where firms used recent recessions as an opportunity to fire workers, invest in computers, and not hire them back, which explains high unemployment. For example, nowadays when you go to supermarkets, you talk to a self-checkout machine instead of a cashier. You talk to a computer instead of a person. And a lot of people have looked at the effect of computers on the economy, but the innovation in my dissertation was this interaction with the recession, that firms use the recession as an opportunity to fire these workers. And then I also show that there's nothing peculiar about computers, that this replacement of workers with technology happened in the past. For example, electricity earlier in the 20th century. I used original data from the US for concrete firms during the 1930s, and I compared regions with hydroelectric power and coal power. Hydroelectric power was very good from the start. So regions like California benefited from cheap electricity, but the price of electricity was constant. Coal power, such as New Jersey, the firms in those regions had initially more expensive electricity, but the price of electricity was decreasing. So I have these two regions, one where the price of electricity is constant, another where the price of electricity is falling. And then I look at the decisions of firms in those regions. And I find that concrete firms producing this output of concrete, they fired workers, they increased labor productivity, and they increased the adoption of electrical machinery. So here's an example. Instead of producing concrete by hand, they just started mixing it with electricity. Which says that also with electricity, as well as with computers, firms fired workers, invested in electrical technology, and didn't hire them back. And then the question is, what happens to these workers after they lose their jobs and they are replaced by electrical machinery? Do they find a better paying job, such as administrative work? Do they find a lower paying job, such as farming? Or do they remain chronically unemployed? And to answer this question, I have the panel of all of the US population from 1900 to 1940 with their occupation and their wages, so I can follow them over time. And this is what I'm working on now, and I'm using the uneven development of the electricity grid throughout the US to trace exactly the effects of occupations and wages for these people after the substitution of it by electrical machinery. So these parallels suggest that, as Mark Twain said, if history doesn't repeat itself, it certainly rhymes. And by looking at the past, we will have a better sense of what may happen to the future of jobs after the current wave of technology adoption. Thank you.
Perfect. Well done, Miguel, representing postdocs as well. So, next up is Henrika. Now, Henrika is from the pathology department, and when you see the slide, it will be amazing. It's a really good theme. He's quite good looking as well. And her presentation is Die Another Way, a novel cell death mechanism in the breast. Over to you.
Cheers for that. So I'm going to be talking about an organ very near and dear to my heart, both literally and figuratively, the breast. And I'm going to do that while utilizing some really bad James Bond puns. So my apologies in advance, you've been warned. So why do we study the breast? Well, it's actually a really fascinating organ because it develops mostly after you are born, not beforehand. And there's a lot of growth happening in the breast during especially puberty and pregnancy, but there's also a lot of cell death involved, which is something that you might not have known about. Because while James Bond teaches us diamonds are forever, well, breast cells actually aren't designed to last forever. They die off after breastfeeding because the milk-producing cells are no longer needed until the next wave of pregnancy. So there's a massive wave of cell death that happens after breastfeeding has stopped. And that is what I study in my PhD. Now it was assumed that the cells die in kind of like the mainstream healthy cell death way that we know about, which is called apoptosis. And to prove this in the lab, we genetically engineered mice to remove the proteins essential for apoptosis in the breasts. And then we looked what happened after breastfeeding to the milk-producing cells. And they still died. They die another way than what we thought. So if it's not apoptosis, what happens to these cells? Well, we found that there's a large increase in the proteins involved in degradation, kind of waste removal in the cells. And they're localized and active in a place of the cell where they're not supposed to be. They're kind of playing a little bit of a villain role in our game here. And when I remove these proteins genetically from the breasts of mice, then the cell death after breastfeeding no longer happens. So that proves to me that these proteins are involved in this cell death pathway rather than it being the apoptotic pathway. Now, the more we understand about this type of cell death, the closer we get to a new license to kill breast cancer cells as well. Because all jokes aside, there's one woman diagnosed with breast cancer every 10 minutes in the UK alone, on average. And we are developing all kinds of new ways of treating breast cancer. For example, in this innovative way of trying to utilize the normal way that cells die in the breast to maybe try and harness that knowledge and kill breast cancer cells that way. So hopefully, in one way or another, this will be a quantum of solace to those affected by breast cancer. Thank you very much.
Great stuff. All right. No, there are a good number of puns in there. All right, so next up we have Anna taking a slightly different turn from the biology department. Now, Anna is from Clare College, and you're going to have to be very patient with me when I say this. Her project is deciphering antibiotic biosynthesis. Getismicin? No. Thank you.
It's a vast improvement. I had to read it last time.
I know. It's really stuck. Thanks.
Thank you.
Over to you.
I'm sorry about the slide. You know, we had such a lovely picture before, and now you're presented with this. But anyway. So not so long ago, we thought that infections were caused by evil spirits and couldn't really do much about it. But now that we know and understand so much more about biology, why is it that we don't have the perfect medicine to treat all the diseases? And it is a known fact that in battle, more people die of infections than from bullets. And we, in our lab, are trying to create the perfect medicine, which will then be able to not only not have any side effects, but also treat any disease that a person can get from just breathing in the wrong bacterium. So my project is on an antibiotic, a medicine called gentamicin. And gentamicin has been described by the World Health Organization as one of the leading therapeutics that needs further work. And that is because it is not perfect. But that is not to say that it's not used in the clinic. It is. In fact, it is one of the most vastly used second-generation antibiotics. So, for example, when the first generation of drugs fails, because the bacteria are resistant to it, gentamicin is used instead. And the reason why it is not perfect is because in 10 to 25% cases, it causes kidney damage, and in further 5% of cases, it causes irreversible hearing loss. And just as a side note, what do you do with the medicine that causes hearing problems? You make eardrops out of it. Please don't buy that stuff. So my project is to generate as many different kinds of gentamicin as I can and to see if it improves its functions, be it either to kill resistant bacteria or to decrease its side effects. And here is the structure of one of the gentamicins, and small changes to the molecule can actually give it new properties. For example, the C2, which only differs from C1a by having a small methyl group added to the top of it, causes no kidney damage in rats at all. So in my project, I have created a vast library of biological machines or enzymes that are able to modify the structure and add decorations or novel groups to it. And currently, I'm in the process of testing to see what machines I can apply to the gentamicin to see changes to it. And from then on, I will also test what the new structures can do and whether or not they have side effects. So, to summarize, why do we not have the perfect medicine? Well, we don't have the perfect medicine yet because I haven't finished my PhD. Thank you.
Well done. Manufacturers of the eardrops maybe don't hope you finish it as soon, but yes. Now we're moving on to our last presentation. That's gone really quickly. So Sophie is representing the Policy Department tonight, a doctoral student, and her presentation is Doctors Under Fire. Over to you.
Ladies and gentlemen, when we think about conflict, we think about bombing, we think about gun. As the previous speaker already mentioned, you're more likely to die of pneumonia and diabetes in a conflict than by a bullet. And the reason for that is that the health sector breaks down. Organizations like the Red Cross find it very difficult to reach patients. And patients don't dare to go to public hospitals because they're invaded by security forces. And oftentimes, they literally rather die than go and risk arrest. It is here where small, underground medical networks of local doctors come literally to the rescue. Small, improvised, they nurse thousands of people back to health. We increasingly rely on them in places like the Ukraine and Syria. However, these small networks face increasing threats. This is a picture of Saif, an Egyptian surgeon. He made it out of the revolution with 20 bullet wounds in his back. Many of his colleagues weren't that lucky. They were killed, arrested, tortured. Bad stuff happened. According to the Red Cross and Doctors Without Borders, this is happening more than ever before in modern history. We just have no idea why these attacks are on the rise. I'm one of the very few in the world studying this question, and it's a very complex one. You're relying on public health, sociology, war studies, international law. It can't get more interdisciplinary than that. The way to do this is to get dirty. You go to the field and you interview dozens of doctors. It isn't easy, and it takes a very long time to gain their trust. What I hope to do with my research is accomplish 3 things. Firstly, I want to know how these networks work. How do they survive, and how do they influence the conflict? Secondly, I want to know why doctors face more attacks in particular conflicts than in others. At this moment, we reckon it doesn't matter whether you're a doctor in a civil war or in a revolution. According to my research, we shouldn't make that big leap of faith. And thirdly, Saif calls himself a medical revolutionary. But can you be a revolutionary and an impartial, neutral doctor at the same time? The Red Cross doesn't think so. My findings suggest otherwise. If we have a better insight in the strategic importance of healthcare in conflict, if we better know the type of challenges these doctors face, we can actually help networks like the network of Saif, and Saif can continue saving people. And hopefully that way, we can also get that perfect medicine to the people who need it most. Thank you.
Excellent. And that's the last one. So, thank God it's not my decision. Our 5 judges will retire shortly to make their decision in lecture room 2.
All right. 3, 2, 1. One more sliding. 3, 2, 1. Fantastic.
Over to you.
Good. So I've been asked to present the results of the competition on behalf of my fellow judges, and the first thing I'd like to say is that it was a really difficult decision. We absolutely loved all of the talks. We learned a lot. We found you all engaging speakers, and in this day and age, you can't get anywhere unless you can engage either your grant funding body or your public or anybody really, your clients, your patients, and you all have bright futures ahead of you. So well done to each and every one of the speakers. We can't give a prize to everybody, and what we've chosen to do is announce 2 runners-up and 1 winner. So, our first runner-up is Joost Haasma. Our second runner-up is Christina Farley. And the winner on this occasion is Henrika Reisman. I hope you might concur with our views, but I fear that might not be the case. But we did come to a definite majority view. So thank you very much for letting us come and hear you and to judge this event. It's been very, very good. Thank you so much.
Now, we're finishing up. I should just do some thank yous. Thank you to our fantastic 5 judges. I didn't lie when I talked about your careers. You are truly... I look at you and I need to go and plan my career out very, very carefully. So thank you so much. And I really hope you join us at the GU for drinks, because I think it'd be a fantastic opportunity to talk to you and find out more about your research, your background, and where you are today, because it's amazing. Thank you to all the participants. The fact that you got this far is incredible. There were 50 finalists who entered, and you all really deserve to be here today. Your research is incredible, and I hope you find the recording and the video we're going to post really useful. It's something you can take with you in your career, and if you do end up at another institution, you can start one up there as well. So thank you to Evian as well. She runs out the door, the president of the GU, for helping put this together. I was doing this single-handedly at the GU, which is incredibly stressful, and Evian has been a tremendous amount of support. She doesn't get enough thanks. Thank you so much for helping out on this. You're brilliant.
And I'd just like to say 2 things. One is, don't plan too much, because grabbing the opportunity might be the making of your career when it comes. And the second thing is, you've done a fantastic organisational job, and no one has thanked you. So, on behalf of everybody, I'd like to thank you.